दुनियाમાં હતું તાને રે.
Hello students, in this module I'll be talking about gene expression and gene regulation. Look, listen and learn. So, remember what I've shown you above me is basically cells, right? These are living cells. One is eukaryotic, the other is prokaryotic. Remember, in a living cell, you basically have gene. And what is gene? You know it is a segment of DNA. Now this gene basically undergoes a process called as transcription, you know, and it makes what? It makes a molecule of RNA, which is a single-stranded molecule. Now this RNA then undergoes a process called as translation, and you basically make what? You make a protein. Remember, proteins are long chains of amino acids. Now in a cell, right, you have two different types of proteins. Either they are structural proteins or they are any other active protein. For example, enzymes. Now all these proteins are ultimately involved in performing metabolic activities, right? Now how does the regulation of gene expression take place? Remember, regulation of gene expression could either be at the level of transcription, okay, it could also happen after transcription, that is called as post-transcriptional regulation, or it could also happen at the level of translation, right? Here I will throw light basically on the transcriptional control of gene expression. Okay, now what you see here once again is a prokaryotic cell. Now in this prokaryotic cell, when the gene forms a molecule of RNA, it is called as transcription. And regulation at this point will be called as transcriptional regulation of gene expression. Now remember, the regulation can be controlled by several metabolites which could be found either outside the prokaryotic cell, so these are called as extracellular metabolites, or they could be found within the prokaryotic cell, therefore they are called as intracellular metabolites. Now both these type of metabolites will help in regulating the gene expression either by initiating it or by inhibiting it. You got that right. Is that okay? So extracellular metabolites or intracellular metabolites regulate gene expression by initiation or by inhibition. Now remember that in a prokaryotic cell, the regulation of gene expression is done with the help of these special units which are called as operons, right? Now what are operons? By definition, operons are a cluster of genes with related functions. Is that okay? And typically operons would transcribe usually a single mRNA molecule. In a bacteria like E. coli, there are about 260 genes that are organized or groups into 75 different operons. Is that okay? Moving on. Let us see a structure of a typical operon. Remember, an operon is a unit of gene expression and regulation and is further made up of two units. The first unit is a structural gene also called as cistron. Remember, this is the part that needs to be transcribed. Now whether the cistron will be transcribed or not will be controlled by control elements, which include promoters and operators. Now let us see where are all these located on a typical DNA molecule. So what I show you here once again is a DNA molecule. Now remember, on this DNA molecule there are certain genes which are called as structural genes, or they are also called as cistrons. You get this right. And what do cistrons do? Cistrons basically code for proteins, rRNA and tRNA. Is that okay? Now how will the cistron get transcribed? For that we require another gene which is called as the promoter. Remember, promoters are areas where RNAP or RNA polymerases will bind so that they can carry out the process of transcription. You got that right. Is that okay? And now remember that between the promoters and the cistrons or structural genes, you find another area or another gene which are definitely called as operators. What happens at the operators? On the operators, you have basically another type of protein that bind, and these proteins are called as as repressor proteins. Remember, very importantly, regulatory genes are the ones that are responsible for the formation of repressors, which then interact with the operators. And we will see that in the next module how it really happens. Is that okay? So in this module, I've talked about gene expression and gene regulation, and I've put forward the idea of an operon. In the next module, I will specifically talk about the structure of the lac operon. Till then, thank you and take care.